Welcome to Lecture 9. Lecture 9 in general is called Microbial Control, but there are two parts to it, and that's why there are two sets of PowerPoint slides and two YouTube playlists. The first is about all the types of microbial control we use outside of infectious disease. So when we're cleaning a surface, or when we're sterilizing a surgical site, or when we are sterilizing medical equipment, things of that nature, that's microbial control. The second set of slides is about antibiotics. So it's specifically about the antibacterial chemicals we use to treat infections. So we will see those in the next set of slides. So we start this set, microbial control, with some important terminology to know. Some of this you've seen before, like infective dose or infectious dose. This is an important thing to know. If we can know this, we can know whether we are safe or whether our cleaning methods keep us safe. Because the infectious dose or infective dose, they mean the same thing. That's the number of cells needed to cause disease. In some cases, it's lots, like Vibrio cholerae causing cholera. In others, it's like 10 cells or 10 virus particles of norovirus can get you sick, but you need 1 million or 1 billion Vibrio cholerae cells to give you cholera. And so then the question becomes, really, how do we take bacterial populations and decrease them below the infectious dose? How do we take them below the doses we care about? If we can do that, we're safe from infectious disease. If we can always clean every surface to below the infective dose, we are safe. So this is kind of how we think about cleaning and disinfection. So here's some more terminology that I'm going to use a lot. Sterilization is a big thing. This is where we've removed all life. So something is sterile if we have killed every single cell or removed every single virion from the thing. So anything we inject into a person should be sterile. There shouldn't be a single cell alive. There shouldn't be a single virion. If there is one cell or one virion, it's not sterile, okay? And so this is often the goal of the kind of disinfection processes we're talking about here. When we're trying to control microorganisms, in many cases we're trying to sterilize something. Many technologies we use, many products we use cannot achieve sterilization. And that's usually okay, because in many cases, the infective dose is more than one cell, so we don't need to kill every single cell. But in some cases, we want to, like if we're going to inject something. A rare term that we sometimes use is sterilant, and this is anything we use that achieves sterilization. Disinfection is the broad term for destroying most of the cells. We also use it generally when we're not being specific. So I'll say disinfection if I'm talking about any process for removing microorganisms, whether or not it can cause actual sterilization. And so a disinfectant is going to be any product that does disinfection. So any product that kills microorganisms or kills all microorganisms. And specifically, this would be on an inanimate surface. So we're not going to use a disinfectant on skin. We're not going to say we disinfected a surgical site. Someone might say that, but that would not be technically correct. So regarding sterilization and disinfection, here is a concept map of the different technologies of microbial control. We have physical things we do, like heat and radiation, different types of those, chemical agents with gases and liquids, and then mechanical things like filtration. And this is too small for you to see, so you don't have to see this. What I want you to pay attention to is that as we subdivide them, here we have physical agents, heat and radiation, and we have dry heat and moist heat, two different types of dry heat. this ending thing here tells you what it can achieve. And these can both achieve sterilization, so that's black. Whereas moist heat, like boiling water, cannot sterilize. It can only disinfect most of the time. And so it doesn't get that black seal of sterilization. So if you look through this at physical agents, some of them do not achieve sterilization. Some chemical agents do not achieve sterilization. So that's an important thing to know. So here are some other terms we're going to see. Lots of different words that end in "cidal," so microbicidal or bactericidal. This is anything that kills whatever's at the beginning of the word. So microbicidal kills microbes, bactericidal kills bacteria, fungicidal kills fungi. And we could compare that to bacteriostatic or microbistatic. This is how you would label a chemical or process that inhibits growth. So it doesn't necessarily kill the cells, it doesn't necessarily kill the bacteria or the general microbes, but it prevents them from growing. You'll see a lot of this in antibiotics. There are some antibiotics we take that don't actually kill the microorganism, but they prevent them from reproducing, so their populations can't grow. And in some cases, they can't damage us. And the key here, the key here is that if you take away a bacteriostatic chemical, the cells are still there, they're still alive, and they'll start growing again. So that's a thing to know. Antisepsis is what we call it when we disinfect skin. Notice I said when we disinfect skin. There just aren't enough words to make this perfectly clean. So I do say disinfect a lot, but when we disinfect a living surface, we call that antisepsis. And then decontamination is a very useful word because this is when, instead of trying to kill all the cells or stop them from growing, we physically remove them mechanically, like scrubbing or wiping. And so some of the products we're going to look at later are mainly used for decontamination, and that can be effective in a lot of situations. And yeah, washing is one of these. So here's some more terminology. Asepsis is what we call it when we've removed pathogens from an object or area. So sterility would be the absence of all life. Asepsis is removal of pathogens. And so medical asepsis is what you want at a surgical site. Medical aseptic technique is what you do to prevent spread of microorganisms from one patient to another or into a surgical site. Aseptic technique in the laboratory is what you learn in a microbiology class. When you use an inoculating loop to do the streak plate technique, that's an example of aseptic technique. That's controlling microbial populations, moving them from one place to another without contaminating them or anything else. And then the last two terms we have here are surgical asepsis, and this is when, as you can see, around a patient who's being operated on, they keep everything very clean, and so everything touching the patient should be sterilized, and they know there shouldn't be any pathogens to get into the patient. And then sanitization is when they, again, clean, they remove debris. And so sanitization and decontamination can be used somewhat interchangeably. And that's something you will see in different contexts. You'll see these words used differently. It's annoying, but you just have to be ready for it. And whatever field you go into, use the terminology the way the people in that field use it, and you'll be fine. So with that, we're going to move to the next video, which starts with modes of action of how these technologies work. How do they kill or stop microorganisms? So I'll see you there.